Botulinum neurotoxin and its cell signaling pathway. Botulinum neurotoxin is produced by the bacteria Clostridium botulinum. It can be found on plants and in the soil, water, and animal intestines. It produces 8 different exotoxins: botulinum neurotoxin A, B, C1, C2, D, E, F, and G. They consist of single polypeptide chains and are one of the most poisonous biological substances known. However, it is commonly used to cure a variety of muscle-related issues, for example, focal dystonias, which deal with involuntary muscle spasms, and spastic movement disorders, which involve muscle contractions and muscle stiffness. They can also be used for headaches, hypersalivation, hyperhidrosis, and some chronic conditions that don't respond well to other treatment methods. It is also used in cosmetics to reduce wrinkles. For example, it is the main ingredient in Botox. This cell signaling pathway involves a motor neuron and a muscle fiber. Vesicles containing acetylcholine have snare proteins attached to them. Snare proteins are SNAP receptor proteins. Those proteins complex with the corresponding snare proteins on the plasma membrane to form a synaptic fusion complex. The vesicle then fuses with the membrane and releases the acetylcholine into the synaptic cleft. From there, the acetylcholine binds with the receptor on the muscle fiber. This signals for the muscle to contract, allowing movement. Acetylcholine is a neurotransmitter, which is a chemical messenger that carries signals from the brain to the body. It is made up of acetyl coenzyme A and choline, which can be found in the food you eat. It controls muscle movement by binding to a receptor. Through this mechanism, it is able to regulate heart contractions, blood pressure, move food through the intestinal tract, and release adrenaline. However, a buildup of acetylcholine can cause muscle paralysis, and a deficiency of it is associated with Alzheimer's disease, Lambert-Eaton myasthenic syndrome, and others. The binding of the acetylcholine signals for sodium channels to open, which then signals for the L-type calcium channels to open. This causes ryanodine receptors to release calcium that activates the contraction. Afterwards, calcium is pumped into the sarcoplasmic reticulum, which promotes relaxation. Botulinum neurotoxin interferes with the pathway by inhibiting the release of acetylcholine, which can cause muscle paralysis. When a human is exposed to botulinum neurotoxin, the toxin is absorbed into the circulatory system through a tissue layer. It then directly and rapidly targets the presynaptic terminal, which is a cluster of axons. It enters the cell through receptor-mediated endocytosis. Once inside, it will cleave into 2 chains, a heavy chain and a light chain. Depending on the type of botulinum neurotoxin, the light chain will affect different snare proteins. Botulinum neurotoxin A, C, and E target SNAP25, located on the cell membrane. The toxin cleaves the protein so that it is unable to function properly. Botulinum neurotoxin C is the only type of toxin that is able to bind to 2 of the proteins. In addition to SNAP25, it also affects syntaxin 1A and syntaxin 1B, another protein located on the cell membrane. Similarly, it cleaves the protein and inhibits its function. Botulinum neurotoxin B, D, F, and G target the snare protein located on the vesicle containing acetylcholine, called VAMP1 and VAMP2. Again, it cleaves the protein from the vesicle. Because of this, the vesicle cannot bind to the membrane and cannot release acetylcholine. A decrease in acetylcholine will lead to muscle paralysis because the muscle fiber will no longer receive signals from the neurotransmitter. In excess, this can lead to botulism, a rare disease caused by the toxin. Since it causes muscle paralysis, it makes breathing difficult and can often lead to death. Other symptoms include muscle fatigue, difficulty blinking or swallowing, and nausea. Therefore, it is important to properly preserve food and treat wounds, as they are 2 of the most common ways that the bacteria is introduced to the body. Luckily, the paralysis is not permanent. Research suggests that the original neuromuscular junction is able to regenerate. A new axon grows from the undamaged part of the nerve cell, so the muscle is able to receive a signal and therefore move again. Thank you for listening.